The low-fat dietary consensus of the 1990s is dead. In its place, competing factions promote aggressively opposing rules for what kind of fat belongs on a human plate. Public health guidelines originally instructed people to replace saturated animal fats with heavily processed alternatives, a directive that initiated decades of confusion over dietary lipids. Today, the internet presents an equally polarized environment. Industrial seed oils are routinely characterized as toxic poisons, while extra virgin olive oil is promoted as a flawless medicinal elixir. Both sides of this debate are measuring the wrong things. They base their arguments on surrogate markers, short-term fluctuations in blood vessel dilation, or lipid oxidation measured in a test tube. To understand how diet actually affects cardiovascular risk, we have to isolate the specific biological mechanism that physically initiates arterial plaque. That mechanism is apolipoprotein B, or ApoB.
According to the European Atherosclerosis Society, cardiovascular disease literally cannot begin without the accumulation and retention of ApoB-containing lipoproteins within the artery wall. As this model illustrates, every single atherogenic particle in your bloodstream carries exactly one ApoB protein molecule. Counting ApoB gives us the exact number of plaque-building particles in circulation. Therefore, evaluating any dietary fat requires a singular metric. If a specific fat does not lower the number of circulating ApoB particles, it cannot stop the progression of cardiovascular disease. A prominent theory circulating online asserts that the omega-6 linoleic acid found in refined seed oils creates a state of chronic systemic inflammation. The hypothesis relies on a specific biological pathway. It suggests that humans consume large amounts of linoleic acid, which the body then converts en masse into arachidonic acid, a precursor to inflammatory compounds. When clinical researchers actually test this pathway in adult humans using stable isotope tracers, the results contradict the inflammatory model. This chart maps the actual metabolic fate of linoleic acid. In human tissue, only 0.3 to 0.6% actually converts into arachidonic acid. The vast majority bypasses the inflammatory pathway entirely and is instead burned for energy or stored in fat tissue. Clinical interventions reveal a different physiological response. When patients replace saturated fats with polyunsaturated options, like safflower oil, the liver is forced to upregulate its LDL receptors. These highly active receptors pull ApoB-containing particles out of circulation, directly reducing the total burden of plaque-forming lipoproteins in the blood. Across randomized controlled trials, increasing dietary linoleic acid has no significant effect on circulating regulatory markers. Instead, it yields a predictable ApoB-lowering physiological response. This brings us to extra virgin olive oil, a product frequently exempted from the skepticism directed at other fats. We need to examine if its reputation holds under the same clinical standards. Vascular biology has documented what is known as the olive oil paradox. In controlled testing, healthy subjects who consumed a high-fat meal derived from olive oil saw their postprandial endothelial function, a measure of blood vessel flexibility, drop by 31%. There are published trials claiming that the polyphenols in extra virgin olive oil preserve this blood vessel function, but the underlying data requires close scrutiny. The majority of trials reporting a unique cardiovascular advantage for olive oil are funded or materially supplied by the olive oil trade industry. Independent tests examining these same endpoints frequently return null or mixed results. The physiological issue is not necessarily the specific plant the fat comes from, but a structural property both seed and olive oils share. They are extracted liquids. Mechanical and chemical extraction strips away protective cellular walls, dietary fiber, and intact micronutrient matrices, leaving behind a highly concentrated calorie source. Without that physical barrier, the purified liquid absorbs rapidly in the upper gastrointestinal tract. This swift absorption creates a sudden postprandial surge in circulating triglycerides, triggering transient vascular oxidative stress. The metabolic burden stems from the form of the food. Extracted, calorie-dense liquids bypass the digestive pacing that the body's vascular system evolved to handle. We must maintain clinical context. Long-term trials confirm that replacing saturated fats, like butter, with unheated liquid plant oils still effectively lowers circulating ApoB and reduces overall risk. Coconut oil is a notable exception. Because its fatty acid profile is overwhelmingly saturated, It entirely lacks the ApoB clearing properties of unsaturated plant oils. To completely bypass the physiological stress of extracted liquids, we must look to fats consumed exactly as they exist in nature, within intact, whole food matrices. The clinical evidence favors obtaining dietary fat from whole tree nuts, like walnuts, unpressed olives, intact seeds, and avocados. Intact plant cell walls and dietary fiber form a robust physical barrier that digestive enzymes struggle to penetrate. This restricts access for digestive lipases, forcing a slow, timed-release absorption of lipids, preventing the sudden postprandial stress seen with extracted oils. This physical format delivers a compounding benefit. You receive the precise unsaturated fatty acids necessary to lower ApoB, paired with the structural volume and fiber required to signal satiety and maintain metabolic balance. The data reveals a clear hierarchy. Optimizing cardiovascular health does not require pledging allegiance to a specific factory-pressed oil. It requires returning to the whole, structured foods the human body is equipped to process.